But we do need to look into the crystal ball because it is looking like algal blooms are not going away. And don't get me wrong, we don't want the algae to disappear completely. They are very beneficial to us. But we do need to control the ones that are more harmful to us. And so just a little bit of background on myself and on my colleague, Paul Frost. Together, we've published over 200 papers on water quality, algae, and limnology, and we've trained over 100 students. What we do is we run a monitoring program just south of you, so the Kawartha Highland Lakes. For the past 9 years, we've been monitoring those lakes. And in addition, we've been monitoring streams and rivers in southern Ontario. And we've been working quite a bit on Lake Erie, in particular, the Western Basin, where they experience annual algal blooms. So we've been thinking about algal blooms quite a bit. And the future is not looking very good. So as the previous speaker touched upon, these are some of the issues that we need to worry about. This picture won the best picture of the year by the National Geographic because they said it looked really pretty with all the shades of green, but this is not pretty. This is not something that we want in our lakes. And these are some of the causes that have been put forward to explain why the lakes are turning green. Flooding, for example, can bring in from the catchment into the lake nutrients that are then taken up by the algae and can produce harmful algal blooms. Poor fishing practicing, overfishing can get rid of the top predators, and this can create a cascade where there are not enough sufficient grazers to control the algae. So this is another reason why we may be seeing algal blooms. We talked about the loss of winter and warming. This could be another reason for seeing algal blooms. And so we are now discovering these are all factors that interact together to create the crisis that we're in right now. And I'm repeating a little bit what was said earlier, but obviously this is a photo from the Toronto Harbor. This is not something that we want to see in our nice blue lakes. We go to our cottage, at least those of you who are lucky to have a cottage or to live on a lake, and you want to just get up in the morning and enjoy the nice blue, clear, or brown because it's the Canadian Shield water. But certainly we don't want the slimy green. And because of all these other issues that we get from algal blooms, including loss of biodiversity, unhealthy fish, loss of oxygen, and poor drinking water, we really just don't want to see that. And my colleague, Jenny Winter from the Ontario Ministry of the Environment, Conservation and Park gave me this slide many, many years ago, which shows the increase of the reported bloom reports that the ministry was recording. And she published this in a paper, and this was subsequently followed up by a different paper, which was presented earlier. So there in the hashed blue line here, this is the Jenny Winter data that I showed you earlier. And what this Favreau and co-authors did is they expanded this list to include all of these lakes in Ontario. So this is massive spatial extent of confirmed cyanobacterial blooms reporting, and especially reported for the first time. So A is the number of blooms being reported, extended from the Jenny Winter data set. You can see that they're increasing. This is still ongoing. And in particular for lakes being reported for the first time, this is quite a dramatic thing to be recording, especially in lakes that we thought were clean. So the question, of course, is why? Why is this? And to reaffirm one of the questions, I think it was Peter asked or made a comment that we need more research and we need to do it at a bigger scale. I couldn't agree more. And we're here to give you a little bit of a sale pitch to convince you that we need to understand why this is happening. So I'm going to say that the number one culprit to explain his algal blooms is still phosphorus. And I cringe every time someone talks about the paradigm and we need to move beyond the phosphorus. If we don't have phosphorus, we won't have the algal blooms. And I have to explain the difference here with what the ministry is reporting through the Lake Partner program, which is total phosphorus. This is a form of phosphorus that includes the phosphorus that's in the algal cells, so particulate phosphorus, and includes the bioavailable phosphorus, including that's organic, but also that's inorganic and that's readily available from algae. The bioavailable phosphorus is the type of phosphorus that is really difficult to measure because the algae are really limited by this. And so as soon as there's a little bit available in the water, it is taken up right away. So normally when we as limnologists go out and grab a water sample and measure it, we want that number to be 0. And so it is usually 0, but it does not mean that it's not there. So we don't have the sophisticated methods yet to be able to detect it in the really small scales that we need to detect it to fully understand algal blooms and its role. So I just wanted to throw that out there and explain that because, of course, you know, and don't get me wrong, the Lake Partner Program is a great program, but with a measurement like total phosphorus, it does not give us the complete picture that we need to fully understand algal blooms. And this is something that we've known since the 60s. David Schindler was my PhD advisor. He's the one that's photographed there. And I did my PhD at the Experimental Lakes area. And I'm sure they would be pleased to hear about doing an experiment, which includes alum or whatever. But that hasn't changed. That has not changed. Phosphorus is still what we need to have in excess to have an algal bloom. Paul, can I ask you to take over?
Sure. Hi, everybody. I'm Paul Frost. I'm also a professor at Trent University. And I'm going to just talk a little bit about why phosphorus still matters and kind of like the background for why, you know, that's probably, we're not going to move away from that. But I think what we're coming to realize is that there's complexities that control its delivery and its access to algae. And that's a lot of that was captured in the last talk. And I'm going to hit some of the same themes and ideas, but maybe a little more simplistically. My PowerPoint skills are not necessarily there. First thing I would point out is if you really want to know about this problem and why the nutrients are at the center of it, get a copy of this book. It's called The Algal Bowl. It was originally published a long time ago by John Valentine and then was rewritten with Dave Schindler. And it explains the basics of lake ecology and why changing nutrients drives algae. And so if you were to sort of get into the guts of this book, you would see something, or you would learn about something like this. So here in the midsummer in a low nutrient lake, I would call a low nutrient lake a oligotrophic lake, which is characterized by low productivity. So there would be no algal blooms in oligotrophic lakes because by definition, they are not productive. And typically what we would see is low external nutrient loading into that lake during the spring or during the summer. And whatever would be loaded into the bottom of the lake would be captured in that cold water at the bottom and would be relatively capped off by high oxygen concentrations. Next slide. If we move to a higher nutrient lake, one that's more productive, this is what we would call a eutrophic lake. This is by definition a lake which has a lot of growth in it. So that would be where you would see algal blooms. And historically, the paradigm was that would be caused by external nutrient loading. So you can see we add a lot more nutrients into the surface waters. This fuels excessive algal growth. It turns green. And now I think the complexity that we're starting to realize is in the absence of that external nutrient loading, we can instead have a lot of internal nutrient loading, which provides nutrients into the lake in a less direct path, but can still fuel excessive algal growth and lead to algal blooms. One thing I would note before going to the next slide is the idea that nutrients can be internally loaded is not new. We've known about this since the 60s, but it was largely assumed that this was something that was a consequence of previous external high levels of external loading, and then those nutrients would eventually get washed out and diluted. The fact that in lakes that are relatively pristine and don't seem to have any external nutrient loading, that we can have internal loading, is actually the new development and something that we need to figure out. And the other thing to realize is that algal blooms come in a lot of different shapes and sizes and flavors. So there's several species of cyanobacteria and different green algae that will become very abundant and cause blooms. And these algae have different life histories and physiologies. Some of them can be toxic. And so for something like microcystis, it can produce microcystin. And we're starting to realize that the nutrient conditions in the water can drive the production of those toxins. So, for example, there's work going on in Maggie's lab here at Trent with her student Carolina, where they're changing the nitrogen to phosphorus ratio that they're exposing the microcystis to, and then measuring the toxin productions, and it's elevated where there's a lot of nitrogen. And that's because those toxins contain a lot of nitrogen. So when the algae has an excess amount of nitrogen, they find stuff to do with it. And unfortunately, that includes making toxic chemicals that are released in the water and can pose a drinking water hazard. So it's not like we don't want to worry about nitrogen or think about other things, but we need to take a multi-nutrient view. And the other thing before I sort of wrap up is conditions are changing. So all those stressors that Maggie talked about on the first slide, this is in flux. So we're going to see shorter periods of ice cover. We're going to see longer periods of open water. We're going to see stratification periods changing, differences in the duration of mixed water columns in the spring and the summer. And this is going to affect things like oxygen conditions at the bottom of the lake. And we've been working in my lab on some of this to look at how oxygen is depleted and its effects on nutrients. And I'll just tell you, the simple answer to that is it's very complicated and it's really varies by lake. And so we sort of need more information to figure out how that all works. And coupled with these changing temperature conditions, we're going to have changes in nutrients coming in from the watersheds because of things like changes in the amount of precipitation and the timing of the spring flush and stuff like that. And so if you sort of layer all of these changes between landscape changes, watershed changes, climate changes, it may be that a crystal ball won't actually be good enough for us to figure out what's going to happen. So if anything, it is imperative for us to start studying our lakes with a view of this and try to put in place the information that will allow us to break it apart and understand it in the years and decades to come. Next slide. So we have been brainstorming around here, and I think this matches what was in the last talk, which is like this has something to do with lake morphology. So sort of like conceptually, this is how we would look at it, is that you might have a larger, deeper lake which has warm water and is well stratified and cold water on the bottom. The cold water at the bottom may or may not go low oxygen, but regardless, the amount of nutrient loading is probably low, and that whatever nutrients would be released would be isolated from what's in the surface water. So over the course of the summer, you'd be very unlikely to see an algal bloom or excessive algal growth in that lake. Whereas if we go to a shallower lake, and this has to do with the morphology, there may be just greater chance for increased internal loading because short periods of stratification lead to low oxygen. That changes the sediments. Those sediments tend to be nutrient rich. Those nutrients go into the water column, and then the stratification breaks apart, and those nutrients are delivered to the surface water where the algae grow. And I just listed off all of the kinds of things that would, because this is very contextual. So whether this happens or not, probably depends on a long list of things. So I've listed off some of the things that play into this. So lake morphology, the shape of the lake, its direction, whether how fast the oxygen concentrations go down, the volume of the hypolimnion, the presence, timing, and permanence of the thermal stratification, and then ultimately what those nutrient stores are at the bottom of the lake. So sufficiently complicated, and I don't think, in terms of like a general paradigm or a general we don't really have the information to be able to say with any level of confidence this lake is likely to have an algal bloom and this isn't. And what I always say is what appears to be a relatively homogeneous landscape. So, but there are this is a problem that we can break apart, study, and figure out. So Maggie and I have been talking about, go to the next slide. Maggie and I have been talking about how we could maybe bring a larger effort to this. And we have access to money from the federal government through NSERC. And it feels like the time is ripe to try to access some of these funds through a large partnership research grant. And that money could be up to $1 million per year or up to 5 years. Typically, we would apply for a couple $100,000 per year. And this would allow us to fund students, sample intensively, deploy equipment, and to break the problem apart and do comparative studies, do small scale experiments, and that kind of stuff. So for this kind of project, we need partners, and that can be private sector, public sector, or not for profit. So this would be at the stage where we would be trying to find partners who are interested in working on this type of research with us. And it seems like your group would be ideally suited and be well positioned to partner with us on something like this. And I would let Maggie sort of talk more about this if there's questions. Next slide.
Yeah, I think I'm okay to talk for a few more minutes. But this is a program. I'm just going to mention, someone mentioned about how the government should help us. This is a program that has a high success rate if we leverage other types of partnerships, and because there is monitoring data that we can tap into. We can try to address the complexity of all those hypotheses, including winter changing, climate change, lake morphometry. We can leverage we can put all of that together to try to pinpoint what it is that's going on and what it is that's driving the increase in algal bloom in our clean oligotrophic lakes in Ontario. And so I think this is what the slide does. This is what we could do at Trent. So Paul and I have started to think about putting this together and getting a team together and finding the partners. But the collaboration is very important for this NSERC program. But we have the personnel and staff, we have the equipment and facilities, and we can train and we can educate the future generation to do the forecasting. Anything else you want to mention on the slide, Paul, or is that good? Okay. So with that, yeah, this is some of the equipment we can also tap into. We're part of these two big networks, the Real-Time Aquatic Ecosystem Observation Network, or RAON, where we have access to a lot of high frequency equipment. So to do forecasting, to model and to do forecasting, we need access to long-term continuous time series. So to be able to detect if there's an algal bloom that's going to happen or not. Nothing against the Lake Partner Program. That is also very valuable. But this is where we need sensors to be able to deploy in lakes. So it's a little bit like when you're trying to detect if someone's having a heart attack, you need a constant signal. And then you see if the signal is deviating from some kind of mathematical normal. So this is what we would do with the sensor data. we would monitor, and then we would see if the variance starts to increase, or the noise starts to increase in the signal. This is an indication that an algal bloom possibly is about to happen. So this is the types of instruments that we would have access to complete this program. And of course, we are also part of the Global Water Futures, which is based out of the University of Saskatchewan. But through this program, we have access to a lot of expertise and modelers to be able to complete this type of project. And so thank you for your attention. And I try to keep this short so that we have time for questions or for comments.